forward to for a long time, partly because you have to go all the way back to last June where we had our Energy and Clean Tech Series planning meetings. We actually had to have two because we couldn't get everybody at the same meeting. And we have a pretty big committee now. And anyway, we decided when we were talking about the Mighty on the Road program, wouldn't it be cool if we get Angela Belcher to come out and talk to us? And so I took on that mission, and here she is. So this is, like I said, something I've really been looking forward to a lot because, and many of you may know this, but this is a very, very amazing, interesting area of work that she does, and you're going to get to hear about it firsthand. So we're really happy that you can make it, and really looking forward to it. So a few words about Angela. Oh, wait, I need to... There we go. There she is. And you know, Mighty on the Road stands for MIT Energy Initiative on the Road. So this is a program that we've been able to put together where we bring MIT professors and other faculty members out to California, and they talk about their research and go into some depth in some of the research. And I've already seen her slides, and it's just going to blow your mind. So I'm really looking forward to this. So Angela is a materials chemist. She has expertise in the fields of biomolecular materials, organic, inorganic interfaces, and solid-state chemistry. So with this, she could probably be in several different departments back at the Institute, but she's in the Department of Materials Science and Engineering, which is also known affectionately as DMSE. You got an acronym for everything, right? And that, along with Professors Don Sadoway and Jeffrey Grossman, who many of you maybe have already heard because we had them previously as part of the Mighty on the Road program. Now, Angela is not afraid to cross technical disciplines and to try to bridge the gap between the living and the non-living interface, which is a very interesting area. And her primary research focus is evolving new materials for energy, electronics, and the environment, about which you're going to hear much more tonight. Now, as a child, she was very interested in being a physician. So she's still Dr. Belcher, but a different kind of doctor, because she was interested in helping people. But later on, she fell head over heels with molecules and the origin of life. And that led her to her current work, evolving organisms to build materials in an environmentally safe way to benefit society. Her greatest achievement may be, however, political, as she's probably the only person to give President Obama the periodic table, which is driving his technology policy today. Her other public service interest is involvement in public outreach, and that includes K through 12 education, something that's also very strategic to MIT. And even though she's a seventh generation Texan, please hold your applause. You didn't know this, Angela, but your governor came out here a while back and kind of dissed the state as he was promoting the state of Texas. And so there's a little bit of angst about Texas here in California, just so that you know. But hey, listen, that was just him. That's not you. Anyway, but she's also founded two California companies. Cambrios Technologies, that's in Sunnyvale, and Siluria Technologies in San Francisco. So she did pick California as the place to do this work, which is notable. Her academic career and awards are in the program that you've received and also in the flyers that we sent out, so you can read those some more if you want. But as noteworthy, I think, as all the awards she's been given is the wide range of publications that have also reported on her work. And I think that's really interesting testimonial that the work that she's doing is of great interest to a large number of people. So please join me in welcoming MIT Professor Angela Belcher.
Okay, thank you. Thank you, Doug, for that very warm and kind introduction, and I appreciate you coming out today to hear a little bit about what we're doing in my group in MIT in terms of evolving organisms to work on new sets of materials. And so I thought I would tell you today a couple of stories, and I was asked to focus on energy, and so I'll tell you what we've been thinking about in terms of evolving biology, mostly M13 bacteriophage, a virus, to work with a larger portion of the periodic table to come up with environmentally friendly ways of organizing and growing new materials. And so this is work that I say this with my dream team because I have an absolutely fantastic group of graduate students and postdocs and undergraduates in my lab over the last couple of years. And it has been a dream team. And then I work a lot with other faculty at MIT, including Paula Hammond, Gerda Sater, Yitming Chang, Michael Serrano, and Professor Young-Xiao Horn. And I'll mention those collaborations as I go along today. And so the focus of our work has really been, can you give genetic information to non-living structures so that non-living structures can have some of the wonderful properties that life has? The ability to make things at room temperature and room pressure, under environmentally friendly conditions, the ability to self-repair, and to get better as a function of time. And the inspiration really comes from an abalone shell, which I did my PhD work on how abalones grow shells. These are calcium carbonate-based materials. They're basically chalk, 98% by mass calcium carbonate, but 2% by mass protein, yet they have about 3,000 times, they're about 3,000 times tougher than the geologic counterpart. And one of the things that's really interesting to me about abalone shells is when a male and female abalone get together, they pass on the genetic information that says, this is how to build an exquisite material. Here's the DNA sequence that codes for this inorganic structure with all this nanostructural regularity and all these really fantastic abilities. And so that's what I've been very interested in, is how do you get genetic information that codes for a material? Same with diatoms, which are made out of silica. There's at least 10,000 different of these diatoms, and they differ from each other based on their overall shape, but they're all made in ocean conditions, and they're basically little glass structures. And each one of these diatoms has the genetic information that says this is how to build a glass star, or this is how to build this glass-looking petri dish, and so it's all coded. And so when I started thinking about future technologies, we start with the beginning of the Earth, about 4.5 billion years ago. It only took about 1 billion years to have life on Earth. But it wasn't until about 500 million years ago, during the Cambrian geologic time period, that organisms started making hard structures. Before that, there were soft structures. And you think, wow, you have life, you have multicellularity, you have photosynthesis, you have all these really fantastic processes that are happening. Why couldn't they make hard materials? What's so hard about hard materials? Well, it was during this geologic time there was increased calcium in the ocean, increased silicon in the ocean, and now organisms actually had the opportunity to start using some of these structures to build materials. And so that's what we thought, well, maybe organisms just need the opportunity to work with the rest of the periodic table so that now that they can expand their toolkit as well. And so we said, well, what if you could give genetic information to solar cells or to batteries? What could you do with it? This is one of my favorite biomaterials. This is my oldest son standing next to one of his favorite biomaterials, the Triceratops. And I'd like to say anyone who has small children or knows small children know they're incredibly complex organisms. And so you think about training an organism to do something new. We're going to think about much simpler organisms like bacteria and viruses. Can you convince them to start working with new materials, to start making new materials, in this case, a battery? And so in my lab, we're interested in the interface between nanoscale materials and in biology. And we think that it actually has a lot of different possible applications. And we're interested in many different global issues like energy and healthcare and the environment. And that intersection between biology and nanomaterials we think is an important tool to addressing many of these problems. And people here really understand the importance of the nanoscale. It's not just about being small. It's about controlling placements of atoms and how they fit together and all the interesting optical, electronic, magnetic, catalytic properties that come out of controlling atoms on the nanoscale, or controlling materials on the nanoscale. And so some of the areas that we're interested in are solar cells and batteries and fuel, CO2 sequestration and storage, and medical imaging and therapeutics in the healthcare, water purification, which is an area of great interest to us right now, more abundant materials, and clean mining. We think a lot of these opportunities can be addressed at this biomaterials interface. And so how do you start to think about this? Well, if you think about the molecules that nature's given us already, you think about DNA and antibodies, which are proteins, or beta-pleated sheets or globular proteins, or even ribosomes, the molecular machinery that is used to make proteins. They're already on the nanoscale. And I always tell the story that I tried to be a polymer chemist for a while, and it was frustrating because I would go and make polymers, and they would have monodispersity, and I'd have to try to figure out how to. I'm looking at my former 304 students over there. I used to have to try to figure out how to separate them out, but biology usually gives you things in exact units because your proteins are encoded by the DNA, and so when you go and you make a protein, or you go and you make an enzyme, it's always the exact same unit in the way it folds, and so I thought that was a very inspiring place to look for a toolkit to start to control materials, inorganic materials, and so. You can think about viruses or protein cages or antibodies or whole cells or enzymes or small proteins, polysaccharides, nucleic acid as our building block to start growing and assembling materials of interest. And going back to the inspiration, here's some examples of some natural biomaterials. This is the coccolithophorid. It's a unicellular algae. This is a 1 micron scale bar here. This is calcium carbonate-based plates that are made around the organism. And this has actually been a big inspiration for our group for lots of different kinds of materials, anywhere from trying to target cancer cells to enhance oil recovery. This beautiful organic inorganic structure. This is the abalone shell again, and if you fracture this and you look at it in scanning electron microscopy, what you see is that this is a 1 micron scale bar, is that it's made out of these plates, these stack of coin plates, and they stack on top of each other. But they don't stack on top of each other just in these columns. They actually interdigitate and they overlap, and it's this structure that actually helps give it its very tough structure. And in between each individual tablet is an organic matrix. It's coded by the DNA that actually nucleates, controls the structure of this inorganic material. And this is a diatom from my undergraduate research made out of silica. And you can see that it's made out of these porous structures, and there's pores of pores that are all controlled by assembly. And then magnetotactic bacteria, which make small single-domain magnets, iron oxide used in navigation. And so all of these are done under environmentally friendly conditions, under ocean temperatures and pressures, and not adding toxic materials back into the environment. We said, wow, what a great manufacturing approach. What if we could do that with lots of different kinds of things? And the secret to this structure here is there's a couple of secrets. One is you have a lot of millions of years of evolution. The second is its nanostructure. And the important part of the structure is actually the proteins. And of course, the proteins are made up of the amino acids, and these amino acids are 20 major amino acids. They differ from each other based on their functionality. And it's how these amino acids are linked together and how this three-dimensional structure folds together that allows this structure to be grown in a very, very regular way, and in a way that the thickness of the tablets is ideal for this particular application, which is protection of the organism. And if you look at look at the scanning electron micrograph, you see these plates, and in between these plates are these organic matrices. And these organic matrices are the ones that actually nucleate the right crystal structure. Aragonite calcium carbonate comes in 3 different crystal structures, and the abalone chooses the metastable crystal structure. And how does it do that? Well, it has a DNA sequence that codes for a protein sequence that allows it to be nucleated and to grow and to be the major structure. And it actually has no perfect cleavage plane, so it's a much more stable structure. And the way that this works is you have this, what I call this an organic matrix, but it's actually, in this case, a protein. And the protein, if you go back and you think about those amino acid functional groups that are linked together, in this case, these are negative charged amino acids, and in the negative charged amino acids, in this case, aspartic acid, glutamic acid, can grab onto calcium, put down calcium carbonate, start to nucleate and grow the structure. So you can think of these proteins as the ability to nucleate a material, but equally important, you can think about proteins that might be on this particular crystallographic surface that are poisons, that keep the crystal from growing in another direction. And so when I started my own independent research, what my big idea is what if you could do this any material you wanted, or most materials you wanted? What if you could have a DNA sequence that coded for a protein that you could use to grow a semiconductor material, a electronic material, a battery material? And so what if you could do this, find this, this interface between these kind of materials, and how would you do it? And I went through a journey as a young faculty member thinking about how you would do that, how you would give biology the ability to work with different parts of the periodic table. And so here's the periodic table, which I handed out one to you that I give out to the freshmen at MIT. It says, welcome to MIT, now you're in your element. And I say that because when I got to MIT, I was an undergraduate and a graduate student in California, and then I took my first faculty position at University of Texas for a couple of years. And when I got to MIT, I really felt like I was in my element. I found my home. And so I said, everyone that comes here will probably feel the same way. And on the other side is the amino acids with their pKa's. And so I do this because it's really useful information. You need to look up, the pH under which you want to know the charge on your amino acid, or your molecular weight in the periodic table, but also to bring the idea, bring these two somewhat different ideas together. The basic building blocks of biology, along with the basic building blocks of all materials. And so, as it was mentioned in the introduction, when President Obama visited MIT, I got to meet with him. He came and visited a joint lab between myself and Professor Paula Hammond, and I wanted to give him this periodic table. And people might have heard the story before, and I was really nervous the night before because I thought, how do you bring it up in conversation, And so. And so after, he was very interested in our research, and actually I guess it was the Secret Service kept saying, you got to get him to move on, go. And I said that was above my pay level. I don't tell the president to, move on. But so I said, sir, I want to give you the periodic table in case you're ever in bind. You need to calculate molecular weight. And he looked at it and he said, thank you. I'll look at it periodically. So then later that day at MIT, he gave a lecture, and he pulled out the periodic table, and he walked up and said, MIT, they give out periodic tables, what's that about? But he seemed very happy to get it. And so what we started thinking about in my group is how this organism started making hard materials 500 million years ago, but it took about 50 million years to get good at it. And that's a really long time, especially on the 10-year track. And so what we started to think is, how can you do it more rapidly? How can you develop that kind of interaction? And so we looked at the biotech industry, and we looked at bacteriophage. It's this M13 bacteriophage. It's a virus. It's a bacterial virus called a bacteria eater. It's a very common virus. And one thing that's nice about it is that it's very easy virus to work with. It has single-stranded DNA and just a couple of genes, and those genes code for the production of the proteins that make it up. And as a material scientist, I look at this as not really a virus, but as an object that's 880 nanometers in this direction and 6 nanometers in this direction. Again, it's monodispersed. Every time you replicate it and make millions of copies, they're always the exact same length and the exact same diameter. And they have their own sequence that says this is how to make it. And so what we did was we went in and we made modifications in the DNA sequence. And these modifications put random DNA sequences in, and these random DNA sequences code for random protein sequences or peptide sequences. And so you can go in there, gene 3, put a short random DNA sequence in it, and that codes for the random peptide sequence here. So basically what I'm trying to do is I'm saying, I know that DNA is incredibly important. I know it's it's inexpensive, it's easy to manipulate, it's the genetic information that tells this virus it's who it is, right? But at the same time, I know it's the proteins that are really controlling the crystal structure of this material. And I know it's the proteins that are controlling the crystal structure of my bones. And so what I want to do is, by manipulating the DNA sequence, I want to manipulate the protein sequence and find a protein sequence that can recognize something that I'm interested in, that through evolution has never had the opportunity to work with yet. And so let's see if we can build that kind of interface. And so we can go in and make modifications in gene 3, and it's actually, I say it's really easy, but I've never done it myself. My students do it. They go in and they make a library of 1 billion possibilities where you go in and you cut and paste and you add a DNA sequence that codes for a peptide sequence. And now we're going to do a combinatorial approach. We're going to do 1 billion experiments at a time. We're going to just go through and look for proteins that can recognize an inorganic material. And this is how they do it. I talked about gene 3. About gene 3, which is a minor coat protein here. There's 5 copies of this protein right here, but there's 2,700 copies, and I'll show you what it looks like, what the protein structure of these look like here, that surround the DNA, and they self-assemble. There is no assembly required. You have the DNA sequence, they're inside of a bacteria, and they assemble themselves, and they're extruded out of the bacteria, so it's really a self-assembling system. And so we can make modifications in here, or we can make modifications in here, we can make modifications in here. So it's a wonderful molecular tool to be able to try to think about manipulating. So my students go in and they cut the DNA in different genes and add DNA sequences, and so they can make libraries of these major coat proteins, which we call P8, or they can make libraries of these minor coat proteins as well. And so what my lab does then is we take a library and we force that library to interact with the material we're interested in, just like the organisms in the ocean started using calcium carbonate to build calcium carbonate. And so we initially worked with semiconductors and insulators, metals, magnetic materials, conducting polymers, many different kinds of materials. And what we're looking for in this case is not only a peptide sequence, but also a geometric arrangement that will recognize material and stick to it. So most of you throw 1 billion possibilities at it. Most of them won't stick. You may have 1,000 out of those that actually have some affinity for this material. You wash it off using different methods. You can lower the pH, which is why you need your pKa's here. And then you're basically, you select for something that has some affinity for this material. Now, you infect it into its bacterial host because viruses can't make copies of themselves. They need a host. You make 1 million copies. You purify it. Now you have a population that's enriched, a population that has some affinity for that material. Now you actually ask it to bind again under more stringent conditions. And you go through this process maybe 5 or 6 times, although I should say we've invented a new process in my lab in the last couple of months to go much faster. But to find the 1 out of 1 million, the 1 out of 1 billion that can do what you want it to be able to do. We first published this in Nature in 2000, and then we've had many different Nature Science and such publications about this over the last several years, looking for the protein that can do what you want to be able to do. And so what do we want to do? And so this is my students made this, and this is a cartoon of me, and it looks pretty much like me. And this is my collaborator, Paula Hammond, and we have a solar project together where we are interested in spray-on solar and inexpensive solar cells. And so what we want to do is, can you give genetic information to solar cells and batteries and catalysts? And we made this actually for Raphael's inauguration. We put him in the driver's seat here. We drop in replacements for fuels, which is a project that we're very interested in, and my company Solaria Technology works on as well. We have a program that I won't really have time to talk about today that actually doesn't use viruses but uses yeast. And the idea is, can you develop yeast? And this is an idea I came up with by teaching 3034. I don't know if I told you guys about that. But when we were doing enzyme activities in that class, which is, can you develop a way of biologically taking CO2 from a coal plant and converting it into building supplies? And here's an example of a building supplies that we made that's biologically made. We used 2 different yeasts to convert CO2 from a mock coal plant, flue gas, to building supplies that are used as tiles. And so these are regular tiles that are a combination of a biologically derived calcium carbonate and a polymer blend that's made by a tile company. And then the same technology Paula and I developed for batteries we're using for ovarian cancer. We've recently developed a method to find teeny ovarian tumors in animal models by imaging all the way through the animal to try to diagnose things before they become metastatic. And then we have programs in solar fuels and photocatalysis as well. So all of this is based on giving materials genetic information so that you can build new things and hopefully become better as a function of time. And so going back to this is our M13 bacteriophage, this is our toolkit. There's a couple of genes. Those genes code for a couple of proteins. This end here has 2 different proteins, so we can make modifications in this gene and add peptides or proteins of interest over here. You can add peptides or proteins of interest over here, and in gene 8, you can put 2,700 copies of a protein that can be used to nucleate a material right here. So it really is a toolkit for, you can think about assembling things in 1 dimension, 2 dimension, 3 dimensions, and multiple kinds of materials. Now, looking at the M13 bacteriophage again, these major coat proteins are beautiful alpha helix proteins that self-assemble around the DNA. And what we're going to do is just give them a new function, which is to nucleate, grow, or bind a material of interest here. And it's a crystalline material. The virus is a crystalline material, so these proteins are crystallographically related to each other. And the initial idea was to try to translate the ability to template on a crystalline level to an inorganic material. And so, again, for the biochemist fans, I know there's some of you out there, that these are some of the amino acids. This is the reaction vessel where this happens. These are the amino acids that not only do you have the amino acids along the individual alpha helix, but they form a 5-fold symmetry and they self-assemble. And you have the chemistries of these overlapping sequences to play with. And so not only are you thinking about engineering on a single strand, but you're thinking about strand-strand interaction and using those as a way of controlling materials. And so, again, as a material scientist, this is an object that has a great aspect ratio. It's long and skinny. And if you can nucleate materials, you can think about nucleating polycrystalline or materials that may be highly aligned, or single crystalline materials like in this case. And when we first did this, we were able to grow single crystal materials. This was for 2,6 semiconductors. I think it was zinc sulfide and CAD sulfide based materials. And we showed that you could grow polycrystalline materials. This is a TEM image. This is actually dark field TEM image that are highly aligned. By heating at the time, we could grow single crystalline materials. And we were so excited about this. See, this is a 200 nanometer length scale. We said we can grow lots of them. But you can sit and you're counting, we're going 1, 2, 3, 4, how many is a lot. And so, but that was a big breakthrough for us at the time. But now you want to think about scaling to a battery, or scaling to a solar cell, or scaling to catalysis. You need to be able to get bigger than that. And so I'll talk a little bit about that. But basically, through the selection process, here's one particular kind of sequence that we've panned and found that was really good for growing gold. And so you start with gold plus 3. You have your particular virus that you pulled out through your selection. You have a mild reducing agent, and you can make gold nanowires. And so you say, well, that's probably not so surprising. But we make these nanowires with 98% conversion efficiency from gold ions in solution into gold wires. And so it's very, very it's very, very good at making these very nice controlled wires. You can control the length, you control the diameter, and this is just 3 little tubes with different clones in them that we tested from our library. And B is our gold binding sequence. It's a great sequence. We like it because it's reproducible in lots of people's hands. And you can see at the bottom the precipitate that's happened. These are gold nanoparticles precipitating out of solution. And you have another clone that we use a lot for some of our battery work. And you get a little bit of precipitation, and then in a control you don't get any. Just basically it becomes very specific for growing a particular kind of material. And this is some nanowires that we grew that are gold nanowires, and we could control the diameter of them. And then you can do other fun things with them, like start growing second materials on top of them. So this is a gold nanoparticle with a platinum core shell structure, which we used with Professor Shahorn's group to work on fuel cells. So we really got interested in actually placement of 2 materials, simultaneously control over putting 2 materials that may be difficult to grow together. And so we started thinking about this several years before, which is, can you give one virus 2 genes, one to grow, in this case cadmium sulfide, one to grow zinc sulfide simultaneously? Can you start to build these heterostructures on the nanoscale in a controlled way? And this is some of our first attempts for it, where we could actually show these are a collection of viruses, and this is transmission electron microscopy data with elemental mapping, and we were excited about it at the time, but it was kind of a toy. We didn't know what to do with it, but we could grow CAD sulfide and zinc sulfide at the same time. We couldn't control the relative placement of these, though. And this kind of ended up to be the bread and butter of our group, which is, how do you put 2 materials together at the nanoscale under low temperature conditions in a way that you can increase the performance of your device. And so here's the idea of you have a nanocomposite, you have nanoparticles. Let's say these are titanium nanoparticles, and let's say these are carbon nanotubes. You wanted to mix them together. Well, you can physically mix them. You can do chemical modifications on these, and a lot of times the chemical modification on something so small, maybe a carbon nanotube, could decrease the performance of your carbon nanotube. But we said, well, let's use a virus as a mediator. Let's engineer the virus to pick up single-walled carbon nanotubes and then grow titania both. And so you have both of those abilities embedded into your sequence so it can bring these two materials together. And so that's most of what I'm going to talk about today is putting materials together. And so this is basically how do you go from DNA to a functional device. And this is we submitted this for, I think this was a Nature Nano last year maybe. And my students spent a lot of time actually building this beautiful diagram. We submitted it for the cover, didn't get the cover, so I'll show it to you here today. But it has the sequence to bind these these single-walled carbon nanotubes in a really particular way. And then it has the ability to nucleate these titanium nanoparticles, which we used for solar cell applications in this case. And some of the first devices that we looked at were batteries. And we looked at lithium-ion batteries. And it was a very radical idea at the time, which is we're going to genetically engineer viruses to grow and assemble batteries. And so basically in the lithium-ion batteries, you have an anode and you have a cathode. We looked at making materials for anodes, materials for cathodes, and looked at engineering the electrolyte some for these. And we wanted to do it in a biological way. And so what did we think that we could get from it? Well, we thought we could get better interfaces. We thought we could maybe get ultra-thin, lightweight materials, 3D materials, which we're actually just getting to now after we said we were going to do this in 2006, higher energy density. But we also wanted flexible, conformable, textile integratable, and room temperature synthesis was really big for us because we always are looking for doing things in the most environmentally friendly conditions as possible. So we looked at engineering biology to grow and assemble these kinds of materials. And so we started off with M13 virus. Almost everything, the answer to us is M13 virus. We used it to grow cobalt oxide nanowires in this case for anode materials. Then we actually were going to use self-assembly to assemble it into an electrode structure. But we used a combination of selection and rational design to try to make them better as a function of time. And I always say that's one of the things I like best about working on our technology is that you may start out with 1 billion possibilities, and you're looking for a virus that can do something, grow a material, say this case cobalt oxide. But if it can grow a material and it's not a very good material, but it can do it at all, you get anything at the beginning, you say, okay, that's great. I have selection and evolution on my side. Maybe I can make it better. And so that's usually the way that we start. And so in this case, it's let's see what we can get, and then let's try to figure out how to make it better, and then assemble it into a functioning device. This work was by my student Kite, who's was course 3 at MIT, and he's now faculty at Seoul National. And so we basically make these in a beaker, and so a flask, just like you would do in a biology lab. We took the sequences that we grew. We have cobalt material. We make cobalt oxide, and this is looking at a single virus in transmission electron microscopy. I think this is a 20 nanometer length scale. Here, these beautiful little nanoparticles that are nucleated along the length of the virus. And so then we're going to test this as a battery electrode material. We put it on copper. We had a separator and the electrolyte. We used lithium as our counter electrode to test it. And what we're going to measure is specific capacity and cycle. And so I always say this is the first data that we did, but it's actually the first data I saw because I think my students don't show me all the things that don't work at first. But and so what they're doing was they're testing the specific capacity of this material, and they're able to get theoretical capacity of it. But it actually starts to, as you cycle it, it starts to decrease, it starts to fade. So we're pretty excited, become less excited and less excited. But we had a reversible capacity about 600 to 750 milliamp hours per gram, which is about 2 times higher than carbon. And we were pretty excited that it worked at all. And if you say, well, how does it compare to, this is all made at room temperature, how does it compare to making just cobalt oxide nanoparticles by itself at room temperature, and that's what it looks like here. It basically is not a very good battery electrode material at all. And so the virus really is doing something, making it more crystalline, controlling the crystal structure, organizing in a way that you get increased performance. But then what Kite did was had a really great idea, which is he said, well, you want the material to be both a good ionic conductor and a good electrical conductor. How do we make it a better electrical conductor? And so he took one of the sequences that we had isolated that was a good gold binder, and he genetically engineered the virus to be simultaneously a good cobalt nucleating material and a good gold binder, and to try to make a new hybrid material and test that as an electrode. And, so this is a TEM micrograph of the virus that has gold nanoparticles across it, and all the spaces in between that you can see, or where it has the proteins that to grow cobalt oxide. And so the ones that have the gold are supposed to grow gold. And so then he gives it cobalt oxide, and it kind of zips it up, and now you have this hybrid material that's a cobalt oxide gold hybrid material, and it has the gene to grow both. And so then you test that as a battery electrode material, and this is capacity versus cycle again. And this is a first generation battery electrode material, and this is the second generation battery electrode material. So it definitely gets better. And we were pretty excited about this at the time when we published in Science, because it was a very, very different idea, and this was in 2006. But we also wanted to be a self-assembling material, self-assembling on a larger length scale. And so Paula Hammond from chemical engineering, one of my best collaborators, works on layer-by-layer assembly of polymers. So basically you have a cationic solution and an anionic solution. You take a substrate or you spray it, or in this case, let's say you take a substrate and you dip it in the cation, anion, cation, anion, and you can build up these layers, these nano-structured layered polymers. And so then we engineered our virus to like her polymers, and then wanted to use this as a surface to be able to grow materials. And we work together a lot on integrating biology with layer by layer now, and there's advantages like low cost, roll-to-roll processing, tunability, et cetera. And so we developed a system where you have a slide, and you dip it in your polymer, and you dip it in your right virus solution, and you dip it in your battery precursor material, and what you can make with these transparent battery electrodes that look kind of like saran wrap. My students actually stopped giving them to me because they kept losing them. And they actually were pretty intensive to make them at first. But they have these cobalt oxide-based materials. And from there, we went on to be able to make stampable structures, stamping membranes, and stamping viruses, and different kinds of structures based on this solution-based process. So we were pretty excited. And we went to our program officers. We had our science paper in hand. And they said, well, you only made the anode material. What about the cathode material? And so then I did what I always do, which is I go to my students, I said, what about the cathode material? And so I had three very talented women graduate students in my group that set about to make new cathode materials based on viruses. And I'll show you the picture in a minute. But one of the ones they started with was iron phosphate material. And they started with amorphous iron phosphate material because it was actually pretty easy for us to grow biologically. And people had been looking at iron phosphate material, not lithium iron phosphate, but amorphous iron phosphate material, but it actually wasn't proving to be a very, very good material. And so my students went in and they made it, they grew it and nucleated it on the nanoscale and showed they could make these nice particles. And then they used some of the tricks that we learned from before by either integrating making small particles, and either integrating them with silver nanowires, or having one virus with iron phosphate and another one with silver, and mixing them together, and they got better ionic conductivity and a better electrical conductivity by doing these together. And they took this material that wasn't that great of a material, and they took it from 75 milliamp hours per gram to over 100, and then with additional genetic engineering, we're able to take it out to 170 milliamp hours per gram. So that's the idea of, if you can get it to grow at all, if you can start to grow material of interest, then you have all these other handles to try to make it better through additional engineering. And so they wanted to look at improving the electrical conductivities of these materials, and one of the ways is by using carbon nanotubes. You can either use conducting additives like carbon black, or people use multi-walled carbon nanotubes. And my students decided to look at multi-walled carbon nanotubes, which are basically graphene, molecular structure of graphene that's rolled up. And probably not that important right now, but you can the chirality of how these carbon nanotubes are rolled have a lot of important advantages for optical and electronic materials. But to begin with, my students, Yoongjung Lee and Hyungjung Yi, they worked on genetically engineering viruses to pick up carbon nanotubes on one end. Here they are here. They engineered them to pick up single-wall carbon nanotubes on one end and then grow a cathode material on the other end. And the idea was that they'd have a good ionic conductor and a good electrical conductor. They would make a powder out of these for a cathode material and then test these as a battery electrode. And here it is here. And we make all kinds of different battery cathode materials right now. And I brought, I forgot my little LED, but I brought some ones that we've just made in the last couple of weeks, new battery materials using the same idea of that I talked about here. And so here's what they did. They were able to use this minor coat protein to be able to grab onto the CNTs. And then looking at the transmission electron microscope here, this the darker part of it is the iron phosphate, and hopefully you can see these carbon nanotubes that are basically coming off here. So what they're making is a network of carbon nanotubes that are really integrated in with this iron phosphate-based material. And so they made a super highway to of a percolating network for transferring the energy in a short amount of time. And I think I took this other slide out because I was worried about time. But one of the things that was, I always say that this was, at the time, it was the second best day of my life. Now with my second child, it's the third best day of my life. When my students went in and they were selecting these viruses to bind to single-walled carbon nanotubes, and what they found was that the affinity of the protein on the tip of the virus, how well it bound to the carbon nanotube, was actually directly related to the power of the battery. And I said, that's it. That's what I wanted to do my whole life, which was have a genetic switch that can control something completely not related. In this case, the power of a battery. And so that was a very, very happy day for us. I should say that this battery. Yeah, so this battery actually went to the White House for a conference. And I would say the battery was invited, but we weren't. We got to watch it on TV, and it was kind of interesting because we published this in Science again because it was an innovative idea at the time, and it was a very high performance battery in 2009, but the field moves very, very quickly. But so we were told that your battery's going to go to the White House, and so my students were all excited, but that we hadn't been making batteries for a couple of weeks because we were finishing our science paper. And it was mostly accepted, but it had some minor changes. And so I called my editor and I said, well, the president's going to talk about a battery. And I said, but we have this embargo. And they said, even the president can't break the embargo. And so I was a little bit stressed, but we'll go ahead and get your paper through, and because it was accepted, it just had minor changes. But then my students were making the battery, and then they had to put it in the FedEx box, and we built a beautiful little case for it, so you could flip it on and it would shine, and they sent it off. And then later that day they told me, we didn't actually test it because we're out of time, but it's always worked before. And so then I'm getting really nervous, and finally we saw the video of it. We saw it real time, and they flipped the switch on, and our little battery was shining, and it made it through the whole speech without burning out. But so that was a little bit nerve-wracking. But basically, what we showed here, and this goes towards the happiest day of my life, which was these are three different. So this is specific power versus specific energy, and this is three different clones. This first clone has one gene, and that's the gene to grow cobalt oxide. The second clone has two genes, and that's one gene to bind carbon nanotubes and one to grow cobalt oxide. But this is our best one. This is our tightest binder, and you can see that it has better performance. And they all cycle very well. They all cycle fine, but what we were doing was engineering it to become better and better as a function of time in terms of its overall power performance, as well as its capacity and its cyclability. And so it was the difference between these two is how tightly the protein binds the carbon nanotube. And then we were lucky when President Obama visited and was seeing our batteries and asking lots of questions. And So we actually moved on from carbon nanotubes and started thinking about graphene, because graphene was such a hot topic too for many different kinds of applications. And so we started genetically engineer our viruses to be able to bind to graphene and then grow other materials simultaneously. And this material was bismuth oxyfluoride material, which has a high gravimetric energy density material, and we showed that we could get it to bind there, and that we could go do additional selection and additional site-specific mutations to make it actually better. And there's a lot of data. Sorry about that. This is volumetric energy density here. But basically, what we found is that we compared them to carbon nanotubes, and the ones that bound the graphene were better than binding the carbon nanotube, so it was a good choice. And that through the additional genetic engineering, we could actually give it a higher capacity through controlling the genes that were coding for the ability to simultaneously bind the graphene, in this case grow the bismuth oxyfluoride material. So again, taking it to a different kind of material. And one of the areas we're looking at now are lithium-air batteries or lithium-oxygen batteries. And these batteries are more next generation batteries that could be used for longer range vehicle applications. So this is current lithium ion batteries and future lithium ion batteries, and lithium air are one of the promising types of materials for longer range vehicle applications. But there's all kinds of challenges with these batteries, and these batteries work by having lithium ions interacting with oxygen, which is one of the reasons why that it has the potential for this high energy density. But there's problems, and most of the problems have to do with the cathode electrode. And so we looked at this and said, is there anything that we can do? Is there anything a virus can do to help solve this problem? And one of the problems is during discharge, the discharge product, which is lithium peroxide, is deposited on the electrode itself, and it's part of the part that reduces the reversibility, and so you need to be able to have it reverse back. And the other part is as this discharge product builds up, it actually clogs the pores and reduces the catalytic efficiency of this. And so we decided to go and look to see if we could use M13 viruses to be able to solve this problem, or to be able to think about this problem. And we did the usual kinds of things that we do, and we looked at making manganese oxide nanowires and then incorporating a second material into them. And so we had our viruses and worked out ability to make manganese oxide nanowires for lithium-air batteries at room temperature. And one of the things we wanted to do was to provide a larger surface area for this catalyst, and to be able to simultaneously have a larger surface area and enhance the interaction of the material of interest in this system. And so I'm showing this actually for the first time, and my student is showing it in MRS tomorrow for the first time. And what Daehyun was able to do was be able to grow manganese oxide nanowires and make this higher surface area material. And then she showed that this is actually this is just the manganese oxide without the virus. And they're looking at cell voltage versus capacity here. And this is the non-biologically templated material, and the biologically templated material increases it by about 40%. In this case, we think that the biology, what it's doing is just changing the architecture, increasing the surface area of this material. But it was a start, and we didn't want to stop there. And so we started thinking, or I should say, she started thinking about what we could do to improve this. And so we were able to take our virus and grow this biotemplated manganese oxide material. It's a mixed valent manganese material, and then we wrapped it with a little bit of polymer, and this polymer had the ability to nucleate either palladium or gold nanoparticles to try to increase the catalytic efficiency of this. And so it actually makes this beautiful structure. I hope that you can see it here. This is the tip of a virus here, so it's a 100 nanometer length scale, so it's just part of the virus. But it makes these kind of spiky structures that increase the surface area individual wires, and the wires, when you put them together into this three-dimensional structure, increase the surface area even more. And this is the manganese, and then she's putting a little bit of palladium here, 1 to 3% is what she was looking at. And then, this is the new data again. So now this is looking at our biotemplated ones. Remember the non-biotemplated ones were here, our biotemplated ones. And then right here, this is very, very good data of what this heterostructural biotemplated material looks like. And so again, it was overall was about a 45% increase again for this material. And she showed that, I didn't show the cycling data on this, but she showed the cycling data got greatly increased, because what we found out is by making this three-dimensional network of these manganese oxide palladium materials, that we could decrease the carbon content. And so we could make low carbon content electrodes. There were 8% carbon content electrodes, and the carbon decreases the cyclability of the system, because it has deleterious effects with the electrolyte, and she could greatly increase the cycling ability with this low carbon, mostly virus, manganese oxide based material that goes well over 70 cycles, which has been a big problem for lithium air, lithium oxygen batteries. So that's our newest. That's almost where we are. We're a couple months ahead of that, but I can't tell you right now. So we started looking at solar materials as well, and we wanted to look at inexpensive solar cells, and we looked at dye-sensitized solar cells. And so we wanted things that were flexible, that were that we realized they wouldn't have the same kinds of efficiencies, but would be easy to manufacture, and we could do it in an environmentally friendly way. And so we did again. We said, what can biology do for this, or what can a virus do for this kind of system? And these dye-sensitized solar cells or Gretzel cells are biologically inspired to begin with, inspired by how on plants. And so basically you have a dye molecule, and you shine light and you promote an electron. This electron is injected into a titanium nanoparticle, and that titanium nanoparticle works its way through in a random way to a transparent conductor here, and then the electrons are collected, and you have the load, and you have an electrolyte here, which basically is involved in recycling the system or recycling the dye. And so what my students said was, what can we do to improve this system? And so you shine light, you have an electron. The electron it's making its way through to the transparent conductor. Things can happen to it. You can have recombination with the electrolyte along the way. You can have recombination with the dye and not make it to the transparent conductor. And so you have a high surface area and high dye loading, but you have a random electron path and you have a short diffusion length. You can think about making vertical nanotube structures where you have a lower surface area, so you have lower dye loading, but you have a directional pathway. So in this system, you shine light, and the electrons have a more direct path. And so what my students did is they said, well, let's see if we can, we're good at carbon nanotubes now. Let's see if we can put carbon nanotubes in the system. And people had looked at putting carbon nanotubes into the system before, but they were actually getting a decrease in efficiency, not an increase in efficiency. And basically what we wanted to be able to do was to shine light and have your electrons, they would find a carbon nanotube and have a more direct path. So you could still have a higher dye loading, but you could have a more direct path. And we wanted to compare semiconducting carbon nanotubes versus metallic carbon nanotubes as well, because as we found out, it was the metallic carbon nanotubes that were actually decreasing the overall efficiency in what people had tried before. The other thing with this is that carbon nanotubes are very hydrophobic, and they basically, in solution, they will form aggregates and they'll collapse together. And we said what we want to do is we want to pull them apart. We want to pull them apart and then maximize their interaction with the titanium nanoparticles. And so we said a virus could solve this. And so instead of this time, remember how when I talked about the battery electrodes, how the virus would bind the carbon nanotubes on its tips through this minor coat protein. my students said, well, what if you can actually start loading them along the outside? And so instead of using that P3 library, we used that P8 library, that major coat library, and we selected viruses that could, normally carbon nanotubes in solution will have a surfactant on them that will keep them suspended. And we said, well, what if a virus could be a surfactant? Not such a strange idea. If you think about go to your amino acids here and you think about what a surfactant is, and you think, well, a virus can be a surfactant as well. What if we select a virus that can pick up carbon nanotubes and start arranging them around the outside? Which my students, of course, were able to do. And they selected viruses, and these are little dialysis chambers here. And so if it's in solution, you don't have a virus, you have clumping of these carbon nanotubes. And so what they did was very, very clever, which they had these, they did their selection. I don't think I have a picture of this. They made carbon nanotubes, single wall carbon nanotubes mats. They took their libraries and they interacted with them, and they pulled out the ones that bound the best. And then they put them in these little dialysis chambers, and then they try to see which ones would precipitate out. If they precipitated out, they weren't acting as a surfactant because or a good surfactant, the carbon nanotubes were collapsing and coming together. But if they stayed in solution, then we had a chance. And so they found a couple of clones that would keep this carbon nanotubes solubilized. And it ends up, if you go and you think about that sequence of 3,000 copies of a virus along the outside. I'm running over in time. If you go and you think about those carbon nanotubes that are on the outside, there's actually 200 proteins that are bound to each carbon nanotube and are keeping them solubilized. I think I started a little bit late though. As the women in my 303-4 class will remember, the answer is almost always histidine on an exam. Because histidine is a very natural, it's a very amazing amino acid. It's an amino acid that can be charged or uncharged at physiological pH. And it was actually the magic amino acid sequence that allowed us to be able to do two things simultaneously. One, it was actually good at binding C and Ts for pi interaction, but also could act as a switch that allowed us to basically change the pH, bind a carbon nanotube, make it soluble, and then change the pH and grow titania nanoparticles simultaneously and grow hybrid materials. And so I'll just skip through this. This is for the biochemist people in the audience, showing you, yes, a virus can be a surfactant, and the sequences that we came up with were surfactant-like sequences. And here's a virus binding a single-wall carbon nanotube. We make lots of carbon nanotubes, and then they actually have a sequence that can grow titania. You grow titania around them, and so now you have a virus with carbon nanotubes and a titanium over them. Now you can take those, you grow those, you take those, and you put them into an already existing titania nanoparticle complex that's made naturally. And you can put 0.1% biology into this system, and you can increase your efficiency of your solar cell. And so here's what we did. So now in here we have 0.1% virus-based CNTs that are growing titania in this already existing matrix. And what happens is again, here's your electrolyte, here's your dye. Your electron is transferred to the titania, and then it can be transferred directly to the carbon nanotube, and you get less back electron transfer and back recombination, and so you should be able to increase your efficiency. Now, if you put metallic carbon nanotubes in, you actually have additional recombination, which leads to decreased efficiency. And so now you're looking at current density of devices here, and this is adding metallic carbon nanotubes actually is decreasing our efficiency. Without carbon nanotubes, at the time, the best Dyson-sized solar cells we could make was about 8.3%. But then adding our semiconducting ones with the virus, we increased it to 10.3%. And then playing around with the ratio of virus and aggregation, we were able to increase it to finally to about 10.8%. And so what you see here is basically you get a 33% increase from adding 0.1% biology to the system. And so that was another big deal for us because it showed you a little bit of biology could go a really long way. We also said, well, what can we do about increasing light harvesting in the system? And so we looked at improving light harvesting with nanoparticles, metal nanoparticles of gold and silver. And the idea was to, if you start to think about electron collection again, you can make it thinner, which will give you more collection efficiency for your electrons, but then it decreases your absorption because you don't have as many dimolecules. And so we said, well, can you increase your absorption by putting metal nanoparticles, gold and silver, into the system to increase your absorption and to broaden out the spectrum of your solar cell. And we were able to do that by making silver, I think this was a silver paper, with titania over it. In this case, we have really thin photoanodes of 1.5 micron, and this is basically with our nanoparticles and without our nanoparticles, and so we see a bump in here. You can actually look, in this case, we have the same efficiency but less materials using our plasmonic, and then we started looking at what we call panchromic solar cells, which is actually broadening the absorption wavelength by adding these materials. And we did some really fun things by growing either silver nanoparticles or gold nanoparticles or core shells of these, and started to be able to tune the wavelength based on adding these. And then we integrated them into our virus-based solar cells. And then we get, a bump in efficiency again over our original structures by adding a very small amount of these specialized, we call them titania gold, titania nanoparticles into this solution. I thought I would tell you just very quickly a story which I call another role for biology in energy and fuels. And people have always asked me about biofuels, and what the take that I had on this was, okay, well, let's use biology to make inorganic materials, but then let's take biology out of the system and see if we can make new combinations of materials to make fuels or other or organic molecules non-biologically. But you had a template to make the inorganic materials originally, and that's the focus of one of the companies I co-founded called Soluria Technologies, and I was here for the board meeting today, which is coming up with new ways of making catalyst for reactions that are very difficult to do in other ways. And just briefly, what we focused on in the beginning was natural gas, taking natural gas and making ethylene, and through something called oxidative coupling of methane. And the idea was that once you have ethylene, you can go to many different kinds of materials, either drop-in fuels or polymers for many different kinds of applications. And so people have been interested in this low-temperature oxidative coupling of methane for a very long time. People have worked on it for 30 years, but it's been a very difficult problem. And so we decided to put biology on the job and see what you could do. And basically, the idea is to go from methane to ethylene, and this requires a lot. We wanted to be able to have existing infrastructure for this, commercially viable temperatures, and as environmentally friendly conditions as possible. And why is this so tricky? Well, in methane, all the 4 bonds are identical. They either stay together or they break. It's hard to be able to control this in a very easy way. And so by doing combinatorial biology combined with combinatorial chemistry and rapid throughput screening, we're able to find catalysts that are able to do that. And of course, the answer is a phage. Using the phage, we're able to design and select these catalysts that have eluded people in the past. And but it didn't just stop at the phage. It really was an incredible team that were able to put in the biology, the combinatorial chemistry, and the rapid throughput screening to make what we have as 2 active pilot plants now, and the next plant stage is in development. Let's see, I really shouldn't push it anymore here, but let me show you one picture that I'm pretty proud of, which is, okay, energy, but what about other things? And so I'm a material scientist, so everything to me is material. It doesn't, the answer is always a material. And so the same materials that we developed for solar cells, which was using biology to hold on carbon nanotubes, once we got really good at that, we said, okay, we can do this. What else can we do with that? And I'm in the Koch Institute for Integrative Cancer Research at MIT. I was asked, my former boss, Susan Hockfield, asked me to see what I could contribute to that field. And so we decided to use some of the materials that we've been developing to see if we could image very early stage cancer in something called the second window near IR imaging. So this is imaging above 1,000 nanometers. And we were interested in this because it's in this special window that is transparent to blood and tissue. And so you should be able to see 10 centimeters into a person or into an animal. And so we, my students and I, thought about this, and then there was no instrumentation to allow us to do animal imaging, and so my physicist built the instrumentation, and Tyler Jacks, I think, who spoke here before, I guess it was here, but it was maybe at a different conference, gave us some money to get it set up. And so this is some work that we're really proud of right now, that we've been imaging ovarian cancer. And ovarian cancer is a very devastating disease. By the time you get diagnosed with it, it's usually at a later stage. And some of the issues with it are that it's hard to diagnose, and normally you look at it by ultrasound or CT, and the gold standard is about 1 centimeter. And it's also really distributed through the space. And so that's working with Professor Sangeeta Bhatia. She's faculty in electrical engineering in the Koch Center at MIT. We started developing a virus that could recognize and bind to, on one end, colon cancer, and then carry with it single-walled carbon nanotubes for imaging. And so this is actually, we work with surgeons at MGH, the hospital there, and we do surgery. This is actually, you look before injection, you don't see anything. You look pre-surgery with injection of our probes. This is in a mouse model, and you see that these bright spots, there's tumors all over the place. And if a surgeon, a ovarian tumor surgeon, comes in and does surgery without our probes, they remove quite a bit of the tumors, but you can see how many are left behind. And the last image is our guided surgery. If the surgeon, Dr. Na, if he used our system as guided surgery, he could find much more of the tumors. And actually, he could find quite a bit of tumors that were 1 millimeter or smaller. And without the guided system, he just couldn't find them. And so we're excited about developing this for both screening and guided surgery. And we just had our guided surgery system delivered, I think, maybe today, to our system, to our lab, to try to see the next generation imager, to really try to start mapping out more information about these tumor distributions in this particular cancer model. So with that, I will definitely end. I thank you for your attention, and I'm happy to answer questions.
Okay, so the way we're going to do Q&A, we're going to have two people in the audience who are going to be roving up and down the two aisles. And I will point out the people that raise their hands if they want to ask the question, and we'll go cycle back and forth, right? So I think we're going to use these two mics. Joe, is that right? Yes. All right. So Anna, Mark, you want to come on down and get your mics? All right, first question. There, gentleman in the blue shirt.
Okay.
Not that blue shirt, the dark blue shirt.
You can hold on to it. He has one.
Don't worry, Harbo, we'll get to you.
Okay, do I win? Okay, Angela, when you're doing the carbon nanotubes and the iron phosphate, do you leave the virus in there, or do you get it out, and how do you remove the biological material at the end?
That's a great question, and we try to leave the biological material in if we can. So it depends on the application. In the case of the iron phosphate materials with the carbon nanotubes, those materials, of course, not the carbon nanotubes, but the carbon nanotubes are attached, and then the iron phosphate is growing at room temperature, and it stays into the structure. But it's all encased. The biological component all is encased in the inorganic material, and so it functions just like a regular inorganic nanowire at that point. It doesn't break down faster than it would if it didn't have the virus inside. In some applications, we remove the virus. In the applications of our solar cells, we go through a post-annealing step, and the virus is burnt out. We published a paper, I think it was either in 2002 or 2004 in Science, where we actually studied the removal of the phage from the inside of the inorganic material, and we found that with controlled ramping that we could actually maintain the same aspect ratio without the inorganic material. Of course, the material is not 100% gone, but it can be burned out. But because we like to work at low temperature if possible, in some applications it's in there, in some applications it's not.
Okay. So I have a question, really a suggestion about how to align your work with the mission of MIT in a slightly different way. MIT not only seeks to do the kind of innovative research, but to diffuse it as broadly as possible. And I wanted to ask whether you'd be interested in experimenting with open access publication for 2 or 3 years. Because at this point, you have, I think, more science papers than anyone I've ever heard a talk from. And when you publish there, you're doing as much for science as science is doing for you. And I realize there's some, risk associated with it still, and your students aren't in the same secure position that you're in. But I would really urge you to experiment with that in a public and open way. I think it could be showing the kind of leadership and interest in practical diffusion of all the work you're doing as widely as possible.
Okay, so I definitely learn more about it. Yeah, I think that this is gonna be a hard question, I'm afraid.
Well, I have a question about your work with carbon nanotubes, and I know you mentioned that nanotubes of different chiral vectors have different electronic properties, and I was just wondering if your viruses are able to tell the difference between.
Yeah, that was my dream and the dream of Rick Smalley from Rice. He approached me about that many years ago, and we were going to do that as well as lots of other things. And and we tried it, and other people have worked on done beautiful chiral separation with DNA, and so every couple of years we come back to that because we weren't successful with it. And initially, when we first started working in it, this is before I even got to MIT over 10 years ago. The quality, you couldn't really get nicely separated carbon nanotubes to start with, and the quality that you get out is always a function of the quality that you start with, and so that was an issue. And then I've had several students start at that before, but we may have the answer to that any day now, based on, and I really hate to say that after having wanted to do it for 12 years now, but we've been working on new screening technology to get much more rare events. And I don't know the answer to it. And if my students know it, they won't tell me right now. But I'm optimistic that we'll get closer to it. There's been so much progress in the field of carbon nanotube separation. You've probably seen in the literature in the last 18 months, and including in our lab, we've worked on bulk separation of different CNTs. But now we have beautifully in our lab little vials of very pure chiralities of CNTs, and we use that now as our starting point to try to do our chiral separation. That, along with our new way of doing much more rapid screening. So I promise you, I'll email you if it works.
Thank you.
Okay, Phil. Yeah, I'm wondering why are Siluria and Cambrios out here?
Yeah. So I asked that myself too. But so when we first started Cambrios, it was in Cambridge, and it was there for a couple of years. And I started it. I'd only been a professor about a year when I started on paper, and I didn't know anything about starting companies. And I started getting all this interest, and I didn't know what to do. So I started a company, and then we didn't know what the technology was. It was a platform technology. And when we were in Cambridge for a couple of years, and we decided to go into the display industry. That's where we're in. That's a display. I didn't talk about it really. It's a transparent conductor replacement. And so when we went to the display industry, and partly because we actually have a lot of venture funding from here, it moved to the West Coast, and it was a really good move because that's where a lot of our partners were in Asia. And I think that when they came here to visit, it was easier to visit us and everyone else at the same time. And so that worked out really well. And so Then when it got time to, so Siluria was a spin out of Cambrios. And I actually was toying with the idea of coming to the West Coast at the time, and decided I was pretty happy on the East Coast. And then when we spun out Siluria started in San Francisco, and again it was an investor thing, and so there it is. But it's a gas company, and so natural gas company, and so it will probably move in other places. Yeah. My next company is going to be in Boston.
You've described using genetic selections in order to find, peptide sequences that bind to whatever target you have. Having looked at the peptide sequences that work, can you now make general principles and not have to go through selection, but design them on your own?
We do, and we had a program on that where we did it basically by doing a lot of experiments to try to tease out the rules for selection. And we did, but of course it's going to be very different for different types of materials. And so we did 2,6 semiconductors, and we did some metals. And from that we were able to rationally design rationally design peptides and show that they could have specificity for different kinds of materials. And I didn't really talk about that today, but we will. A lot of our libraries now that we start with are libraries that were started off from rational design, and so they will be biased libraries. And then from the biased libraries, we go through a selection process again. We do a combination, but almost everything we do now has a rational design component as well as a completely combinatorial selection. But we do have learned a lot.
Thank you. I was interested in the last couple of slides you showed, and I was wondering whether or not the mechanisms that you're looking at here for enhancing imaging could be used for drug delivery or even delivery of something that you could use then in order to focus a therapeutic.
So thank you for the question. So that the question had to do with, we mostly developed this as an imaging technology because that's what we saw a real need. And there's a lot of experts in delivery at MIT. But we had a paper this year in, I think it was ACS Synthetic Biology, where we showed, and it was only in vitro, wasn't in vivo, where we showed that we could, it was prostate cancer, where we could use the phage to bind to a prostate cell, tumor, and then bring with it a chemotherapeutic agent. And we think that we can do a lot more around that. And we've started working with, again, with Professor Bhatia at MIT and doing other clever things like light-induced released or heat-induced release of materials. And we have a new class of particles that we're working with that we think are ideal for a combination of IR and release. And so we are definitely going in that direction. When I get into something, I always ask, can we make a big impact in that? And I don't know the answer to that yet. I'm feeling pretty good about our targeting. I feel very good that our system is very compatible with ovarian cancer because actually in that case both the therapeutics and what from my understanding in ovarian cancer and in imaging are delivered IP. They're delivered directly into the intraperitoneal space, and so you don't have to go through the IV. And this system actually is very good for that. But it's something that we're going to start looking at. We know that through this through coupling chemistry that we can add chemotherapeutic agents and other materials on there. I think we had a paper just accepted this week on imaging infections using basically targeting staph, having the virus target the staph, carry with it a imaging agent, and then we've looked at carrying with it specific antibiotics at the same time. So we're on that road. I'm not sure we'll hopefully we'll be able to make a difference with it.
Larry.
Back to your energy storage. Do you have have you decided where you want to go in terms of what kind of storage you're looking at by application? In other words, stationary storage for grid applications or electric vehicles or handhelds. Yeah. And what sort of cycle life are you achieving, and what are your targets?
So, we looked at, when we first started in this area in 2006, 2007, we looked around, and there's, the competition in the battery space was really intense and getting very big. And we looked at, can we scale these viruses? In those cases, the viruses are the major component of the active material, and so you have to make a lot of it. And so we started thinking about where we would fit in. And so what we looked at were architectural batteries is what we got interested in, which are batteries that are structural and energy storage at the same time. And so we were working with the Department of Defense for micro UAV applications, where the weight and architecture of the little plane is very, very important. And so we have a program where we're making the batteries are the wings of these UAV applications. I always say that if my students could make a battery that for my Prius, but it would be very selfish because it would take a lot of work just so I could have a virus-powered car, which is one of my dreams. But I'm thinking Siluria will take care of that with the virus-powered gasoline-driven car. But so I don't see it's competitive in that market. I see it as more of a specialty in the architectural space. Now, we've only been in this lithium air space for about a year, and we think that we've made a pretty big jump in that. We'll see what people in the community think. Fortunately, we're very lucky to work with Professor Schauhorn in mechanical engineering. She's an expert in lithium air batteries. And she's pretty excited about it. So maybe that's going to be a good break for us. Then we'll have to think about scale in that case. So grid storage, wow, that's big. I think it's a lot of work for a little virus.
And cycles?
Oh, cycles. We never cycle things out to more than 100 cycles or so. I think our best cycle with our lithium air battery is like 70 or something, which is actually really good for a lithium oxygen battery electrode. But like I said, that data that we have on cycling for that is less than 3 months old, and we have ideas of how to make that better. But in the literature, they were publishing 20 cycles for those kind of batteries. Yeah, I understand you want 10,000 cycles.
Right. Yeah. By the way, IBM has a lithium air project.
I know. Yeah.
Okay.
Are you on that project or
no?
Okay. Yeah. So is there data better than ours? I haven't seen the data. I've only seen the press releases. Yeah, okay.
Have you tried to make a abalone shell in vitro?
Yes, I did my PhD on that, and I didn't make a big abalone shell. It was my second Nature paper. Was basically using proteins to control the aspect ratio and grow tablets. I only grew like 10 tablets, but they had the right thickness and the right crystal orientation. And that was the whole inspiration for all my work, which was what I showed was that proteins that were physically isolated out of the abalone shell, you could take them, put them into an Eppendorf tube with the calcium carbonate, and make the metastable crystal structure and start to reproduce that. I never, once we did that. I never saw a reason. My first Nature paper from graduate school was on making flat pearls, actually, which was really fun. Which was trying to understand both biochemically and structurally in time how an abalone shell was made, and it was important for me because I was California. I was vegetarian. I didn't want to kill the abalone, and so I figured out a way to collect its shell without killing it, and kept collecting the biochemistry and put it right directly on the X-ray diffractometer at the same time. And so we did make synthetic pearls that way, and then we made them in the little Eppendorf tube, and published that. But then it was that, and then it clicked into me that, wow, why not something else? Well, let's move beyond calcium carbonate. So that's where I've spent the rest of my career so far. I think there's a student question over here. Yeah.
So I had a question about the plasmatic nanoparticles that you're using in your dye-sensitized solar cells. And I was wondering if those are coupled to the viruses.
So we published a couple papers. I think it was this year, and in those cases they weren't coupled to the viruses yet. Those were made separately. We made the gold or silver nanoparticles, and we put the titanium core shell on them, and then we made the core shell core, however you say it, particles. And then in our latest work, they're actually coupled. Yeah, I don't think that's out yet.
I'm wondering if you have an opinion about the do-it-yourself biology movement, and if you see the amateur or garage biologists being able to tinker with this type of space, or if it really needs to happen in a university laboratory.
I want to do it. I want to make batteries in the kitchen. I think that the biology behind this is very straightforward, I think I'm all about biology for good, I think that it's pretty simple, and I see it as possible. I mean, I think that I have a 7-year-old, and I told him he could have a present recently. He goes, "Oh, what I really want is a 3D printer," and so we're shopping for 3D printers right now, and I think it's the next generation. I think that everyone's going to make things themselves, and I think it's a great movement. Yeah.
Okay, we'll do one more, and then would it be okay to maybe a few people come up afterwards and talk?
Yeah. No,
okay.
So I have a daughter at MIT, is a senior in Course 10B, and she just committed to Cornell for a PhD program in the same field. And I noticed you have 2 young kids, and you're on a tenure track. I'm just wondering, how's that working out for you?
So, it's something I've been thinking about quite a bit because of all the intense media right now about women and professions and raising families. And so I have a 7-year-old and a 2-year-old, and you say, can you have it all? And I completely have it all. I mean, and I have companies on top of it. But you can't have. But it's like it's evolution. You have it. Not every given day do you have it. Every day is chaos. But it's almost like the average of that is really, really great. And so it's completely true. And I think that in terms of academia, it's a really good compatible lifestyle for me. It's been a very good compatible lifestyle with raising a family, because your time really is your own, and we tend to be, like everyone that was here and from MIT, really driven people. But you can, I pick my kids up from school almost every day. It's important to me. I teach in my son's school once a week, and you just figure out how to make it work. But then I can take a month off in the summer if I want, and I can I don't have to ask permission to go teach my son's class once a week. It's just a flexible. I like being the boss. It's about, if you're the boss, I think it makes it a lot easier. And so I think it's a really good profession for that. But I started thinking about that today, and I started going through the list because I was reading the book Lean In on the plane, because people said you have to read it. I thought, I don't need to read that book, but it's pretty interesting and insightful. And I think that being at MIT has been a huge advantage, and I think it's because we've made a commitment to have great child care on campus. My children have always gone to school next to the building next to mine, and I can drop them off and know that they're safe. And before my office moved, I used to spy on my son when he was 4 years old. Out the window, I could see the playground, and I could see him go out with his friends, from my office. And I've been really, really fortunate to be in a place where that's possible. And so I tell women that are on going to look for faculty positions right now, I ask them to look around and make a comparison, see what it's like. Are there women with families if they're interested in having families, and what that's like. And you go around and you look from everywhere, from our president, our former president of MIT, Susan Hockfield, who was a fantastic mom, and to department heads and that went through tenure and rose to the dean or department head while raising kids, and it's definitely possible. It's not easy, but I don't think raising kids is easy by itself. I would say it's way harder than being an MIT professor, for sure. So I think it can be a very compatible job, but I think that it depends on your environment. And like I said, I don't think I could have done this at the university I was before. I think that finding a place that's very supportive of that is important.
I think it's great with all the science that we ended the Q&A session in a personal and human note. So now, Joe, would you switch to the Part PC, please? So we can properly thank Angela. So,
thank you.
Angela, thank you so much. And don't go. As is our tradition, we'd like to present you this bottle of California Cabernet.
Thank you.
It's a Camus 2010, just in case you were wondering. And for the chemists in the group, it's Wine Spectator 92 points. So don't drink it right away, though.
I told my husband about it. He said that he wanted to drink it too. So.
Good, great. Okay, well, if everybody just stay put for just a couple of minutes, I want to go into the other thank yous and the coming attractions, and we have a lot. So first of all, the tour of the California ISO control room. Now, how many people here in this room went to the first one? Raise your hand. Okay, a few. There's still a few seats available on this. This is a fabulous tour. I learned so much about how electricity is managed and the flow of it within the state, and these guys are in control of the whole thing, so it's really neat. It isn't Folsom, so it's a long day, but it's really worth the trip. The next one is coming up. This is really exciting. We call this the world's largest nuclear fusion experiment. The National Ignition Facility is part of Livermore Labs, and they have a facility the size of 3 football fields with 150 high-powered lasers, all pointing at a 2-millimeter target. And hitting that thing and raising the temperature there to over 100,000 degrees. So it's really cool, and I'll be there for sure. This will be our first one. I expect we'll probably do a few more. And one thing about this is, unfortunately, for a few of you maybe, is you do have to be a U.S. citizen to attend this one, but we're working it out with them so that in future we'll be able to do it for non-citizens as well. Now, this is the other one that I'm really excited about, our last of the Mighty on the Road series this season, and we're really, really excited about having the head of the EE and CS department at MIT. So he used to be just a professor, and nothing wrong with that. And we invited him to actually come and do this right at the same time that he got promoted. So we were really fortunate that he decided to do this out of his busy schedule. He is an expert in ultra-low power circuit design, and the connection with energy, of course, is you don't use very much. And we're hoping, actually, that for all those who are really into electronics, that this will be a really exciting presentation in late May. Well, and I got to do this as a tease, okay, because we haven't got it nailed down yet, but many of you probably know the solar airplane, Solar Impulse, is at Moffett Field in the hangar there, and we are trying to arrange an exclusive evening presentation for the MIT Club Energy and Clean Tech Series. So be watching for the promotion on that. It will probably sell out pretty fast, although they say they can handle up to 250. And so we'll have to cap registration at that. But even so, I mean, I just think it's going to be insanely popular. So, and one of the things that's neat about it is that the CEO and co-founder, Andres, is an MIT alum. So a little connection there. So first of all, I'd like to thank Park very, very much. Rob McHenry. Rob, you want to just identify yourself? And And Rob's really in charge of the energy program here at Park. So he's really key person with this. We appreciate his support. Lisa Andreasen was our admin support. Marcy Steiner in the kitchen and outpouring wine. So if you like the food and well, not the wine, I bought that, but the food, that's Marcy's thing. And Joe Sullivan back in the booth there, handling AV, along with our own Hank Magnuski. I'll first of all say a word or two about the park program. So here is how it lays out. There are a lot of things going on in batteries, in solar, in what they call adaptive energy. Not sure what that is, but there's some details, and other enablers in the whole energy field. And there's three ways that Park actually can interface with those of you either here or your friends and associates. One is to businesses that are looking for innovations may be able to find some here that they could license to perhaps start a company around. Second one would be startups that are looking to be incubated here. So Park has become an incubator, and a very important one at that. So that's another way to work with Park. And then the last one, if you'd maybe like to work here and do some of this research in technology, that's another possibility. Anybody personally interested in that, please come up and see Rob afterwards, and he'll be happy to tell you more. And finally, I'd like to thank the MIT volunteers, Hank Magnuski, who's our videographer. Well, he's back in there. You can't see him. So, but he makes the videos for all these events, and we will post this event on the MIT Club website and also on MIT Tech TV. So if you want to go back and see some more of what you heard, take a look at a slide or two on that, or you want to have your friends who missed this opportunity to see it, it will be there. And then finally, just want to recognize our president, Elizabeth Eng, who could not make it tonight, but our classical photographer, Bill Murray. Bill, you raise your hand there. And the volunteers, Poravi Shah, yeah. Well, maybe she had to go back early. That's right. Liz, she had to go back early too. They both come from San Francisco. So Greg Dieguez. Gee, you too? Anna, I know Anna Waldman-Brown is here because she is handling the mics, and Mark Hsu on the other microphone. So thank you all very much. And we do hope to see many of you again at one of our Energy and Clean Tech Series events. Thanks again for your support.